Hey, welcome to the next video. This one is on mitosis. Real quick up front, I should mention that the book treats cytokinesis as kind of a separate event. I treat it as just the end of mitosis, but you do want to keep in mind that cytokinesis, or dividing up the cytoplasm, isn't just having some of the liquid ooze in one direction and some of the liquid oozes in the other direction. You've got things like mitochondria and lysosomes that need to be equally apportioned between the two daughter cells, and you've got more complicated structures like endoplasmic reticulum and Golgi apparatus, which have to be divided up and structurally reassembled in the two daughter cells. So it's a lot more complicated than just a little bit of cell juice goes this way and a little bit of cell juice goes that way. Some other terms that we have to identify up front is first the mitotic spindle. This is an array of protein fibers, so they're like cables of protein that we call microtubules. And they attach to the chromosomes and is what are going to guide the chromosomes to either end of the dividing cell. The microtubules are organized by structures called centrioles, and the centrioles form this larger structure called the centrosome. So the microtubules of the spindle are going to originate at the centrosome and then attach to the centromere, which is on the chromosomes. There we go, I'll even add that in there. So we start in interphase. This is the time in between cell divisions. The DNA is loosely organized as chromatin. You have a fully formed nuclear envelope and these two centrioles within the centrosome. After you go through S phase and end up in mitosis, the first stage of mitosis is called prophase. Here you have replicated centrioles that are moving to either pole of the cell and forming the mitotic spindle. So these are the centrioles within the centrosome, and these are the microtubules. During this time, the chromosome, or the chromatins, I should say, condense into visible chromosomes, and the nuclear envelope degrades or breaks down. So you have some very pretty pictures here of going from chromatin to chromosomes and the beginnings of this mitotic spindle being formed. When you get to the next phase is metaphase. During metaphase, the chromosomes line up in the middle of the cell, and you now have fully formed mitotic spindles that are attached to the centromeres of the chromosomes. The next phase is anaphase. During anaphase, the chromatids separate. Once they separate, we call them chromosomes, and the newly formed chromosomes then migrate to the poles. Then you get to telophase, which is basically prophase in reverse. So in prophase, the chromosomes condensed. In telophase, the chromosomes relax or decondense and go back to being chromatin. The nuclear envelope reforms, and then you get what is called a cleavage furrow, which is just where the two cells start pinching off from each other, and it's at this time, the end of telophase, that cytokinesis takes place. So as it says here, cytokinesis usually takes place during telophase, divides the cytoplasm. And I just wanted to fast forward to this picture because I think it's a funny looking, it looks like a black and white peach, but you get an idea of what the cleavage furrow looks like in a dividing animal cell. And we're not really going to worry about the difference between animal cells and plant cells. We're just going to know the phases of mitosis. So that's mitosis in 5 minutes.